So good afternoon, everyone. First of all, I'd like to thank all the people involved in the organization of this Congress, as well as Alexander and Adam for this symposium on vanilla, which gives me the occasion to talk to you about the genome of cultivated vanilla and with a focus on vanilla planifolia, in fact. So, well, yeah, I'm Quentin Piet, a PhD student in the Joint Research Unit of Plant Populations and Bioaggressors in Tropical Ecosystem, which is a joint research unit between University of Réunion Island and CIRAD. I'm supervised by Professor Pascal Bess and Dr. Cyril Jourda as part of a team focused on vanilla diversity with strong interest, vanilla genetics, sorry, with strong interest on vanilla diversity and varietal improvement. So, well, when we talk about cultivated vanilla, in fact, we mostly talk about three major species, which are vanilla planifolia, pompona, and tahitensis, with planifolia alone supplying more than 95% of the commercialized pods in the world. And so that should be the focus of this talk. And well, this species originated from Mesoamerica, and the thing is that they are nearly exclusively vegetative mode of propagation from this region to their current area of cultivations around the world has led to a really limited genetic diversity, and so to a certain vulnerability to biotic and abiotic stresses. And well, this is illustrated by the fact that planifolia, as well as other vanilla species, are subject to many fungal and viral diseases, such as the fusariosis, also called root and stem rot, that is caused by a fungi named Fusarium oxysporum, and which is one, if not the most devastating disease for vanilla cultures around the world. There are also viral diseases and some pests that cause substantial damages to the cultures also. And on the abiotic threat side, there are also premature fruit drop that has been a cause for concern in the recent years, which is sometimes attributed to global climate change and rise of temperature during the pollination season. But well, that's subject to debate, and I saw that there is a poster outside that tackles some of aspect of these problematics. Well, so the face of all these threats and the inability from these cultivated species to be resilient against these diseases and all these factors, there is a urge for varietal improvement and varietal improvement programs. But the problem is that these programs in their traditional forms can be really laborious and really long, depending on the species it's focused on. And well, it's the case for planifolia and other vanilla, with a total duration of around 10 years between the seeds and the moment when you can begin to evaluate mature pods for their agronomical and commercial value. So there's a need to speed up this process. One of the ways to try to enhance these varietal improvement programs is to better characterize the genomic sequence of these species, as well as the gene that compose these genomes, in order to provide researchers with some insights, and so that they can try to identify some genomic regions or genes of interest that can be involved in some agronomical traits that can interest the sector. So yeah, so let's talk about this vanilla planifolia genome, which is a diploid genome showing polyploidization events. Cytogenetics and flow cytometrics analysis evaluated its diploid size to be around 4 billion of nucleotides, and also showed that so the genome was composed of 16 pairs of chromosomes. But we often observe, in fact, frequent somatic aneuploidy in the cells, with metaphasis chromosome counting often showing less chromosomes than the 16 pairs. And in most of the cases, we observe, in fact, 14 pairs of chromosomes as shown in these karyotypes. So, well, as I said, there's a real need for genomic resources. And it's only in the recent years that the efforts to try to characterize this sequence have been made, with 2 chromosome level assemblies that were published. The first one published by Eising and its collaborators in 2020. So they used the cultivar named Daphna. They extracted high molecular weight DNA from their leaves, from mature leaves. And well, they sequenced it with a combination of 20 long reads, 29 long reads and Illumina short reads to assemble the genome that you can see its circular representation with the 14 pairs of chromosomes on each haplotypes. And if we look at the protein code genes, we can see that they notated nearly 60,000 genes, protein coding genes, and that analysis of completeness showed that it was a good representation of what you could expect for this species. But there are 2 problems. The first one is that there are only 14 pairs of chromosomes that are represented, and only also a bit more than 1/3 of the expected genome size that is represented in these sequences. But well, it was still a huge improvement for the sector, with all this sequence of protein coding genes that allow researchers to reconstruct some metabolic pathway of interest. But well, we can still ask ourselves why we observe such differences with the expected metrics. And in fact, there are 2 main factors that can explain it. The first one is the high heterozygosity rates of this genome, as it has been shown by several studies using different kinds of markers. But the main reason surely is the orchid-specific phenomenon of partial endoreplication that has been mentioned this morning. Which is, as you guessed, a kind of endoreplication, which is itself a really common phenomenon in plants, with more than 90% of angiosperms subject to this phenomenon, and that correspond to DNA replication without mitosis at the end. So here you have got genetic representation with the doubling of the nuclear content at each endocycles. And while this phenomenon is often associated also with favorable agronomic traits like organ development, fruit size, resilience and resistance to some stresses, abiotic or biotic. In the case of orchids, we also observe species presenting a variation of the phenomenon, that is the partial endoreplication, where only one part of the genome will be replicated at each endocycle, while another portion, noted here, which is the fixed portion, will stay in its diploid states. And in fact, if you see here the histogram of fluorescence intensity obtained from Popidium idiom, I don't remember the name, you can see the expected position of each peaks, which corresponds to the polyploidy of the nuclei, which is expected for a whole genome replication at each endocycles, and you can see that there are shifts between what is expected and what is observed in this orchid species. The problem with this phenomenon, is that these regions tend to be underrepresented in the nuclei, and so we struggle to catch them when we try to sequence the DNA, and so that explains why it can be missing in the genome assembly. There are two particularities of this phenomenon that are interesting. The first one is that the proportion of genome subject to partial endoreplication is species specific, with vanilla planifolia and titaenses showing approximately 26% of their genomes that are replicated at each endocycles, while some other vanilla species like Pompona and Mexicana show very low or very huge amount portion of their genomes that are replicated. So, and it varies among other orchid species as well. And the second one, and one which we are lucky, in our sequencing project, is that the proportion of endoreplicated nuclei is also tissue specific in a given species, with some tissues like the nut showing higher proportion of lowly endoreplicated nuclei, so 2C content for E, 8E, than some other tissues like leaves, who show really high portion of endoreplicated nuclei. So that can explain if you don't choose a 2C enriched tissues for the sequencing, you'll find that all this portion of the genome will be underrepresented, and you risk to miss it in your final assembly. So this leads us to the second genome assembly published in 2022, another chromosome level genome assembly, which was published by the VANISEC consortium that regroups French private and public institutes. And so for this project, we sequenced a traditional clone from Reunion Island. We took notes from in vitro plants from which we extracted high molecular DNA with several sequencing technologies. We tested for different assembly approaches, and the best one was in fact obtained with the PACDO Hi-Fi data alone. So the scaffolding was done with optical maps, but also with the help of the information already available with the previous assembly that I just showed you, especially the IC data. We also extracted RNA from various tissues in order to proceed to an automatic structural and functional annotation of the genes of the genome. And well, just another point is that all these data are accessible via a web portal, which also provides comparative genomics analysis tools. So as I said, we choose nodal tissue since it showed a good balance between 2C or lowly underreplicated nuclei and their availability in plants. So because in fact there are other tissues that are like devoid of underreplication, like the pollen, for example, but due to the flowering cycle of the plants, it was impossible for us to obtain sufficient quantities of pollens to extract the DNA and sequence the genome. So here's our result with another circular representation. So you can still see here in colors the 14 pairs of chromosomes. And what we added, in fact, compared to what was previously obtained, is all these unanchored sequences in brown, which represent 2/3 of the genomes that we didn't succeed in anchoring on the chromosomes. And that are characterized by really low sequencing depth in green, high rates of repeats, and very few genes except from some scaffold. So if you look at the protein coding genes content, we see that we didn't really change anything compared to what was obtained from the previous assembly. And what we added was mostly composed of repeats. So we suppose that this fraction of the assembly is in fact correspond in fact to the non-underreplicated regions of the genome of the plant. And well, we see that we are still missing like 20% of the genome in our data, so there's still room for improvement on the structure. So with this assembly, we also try to obtain a first image of the structure of the partial endoreplication phenomenon. For that, we in fact realigned sequencing data from two tissues, so the mature leaves and the nuts, obtained using different sequencing technologies. And we mapped them onto the 14 pairs of chromosomes that we had. You can see, in order to analyze the variation in sequencing depths along these chromosomes, as you can see here, with the colors corresponding to the different sequencing technologies and tissues. And it's allowed us to determine two patterns of the structure, with the first one corresponding to regions for which we observe sequencing dropped for both tissues, regardless of the technology sequence or the sequencing technology used for that. This region are characterized by few genes, low genes, and a peak of unclassified ritual transposon that we need to investigate further. The second patterns correspond here to region where a drop of sequencing depth has only been observed for nodes. It also seems to be haplotype specific, with this drop only occurring in one of the haplotype. You don't have the image since only put one haplotype, but so it's really preliminary results, sorry, but it shows us that we need a more contiguous assembly in order to resolve structural questions, including those on partial endoreplication. So that's what we are working on by combining several approaches, and by keeping in mind that we need to choose suitable tissues for that. So we will use IC data, as well as genetic map, linkage map information that was obtained by Félicien Favre in our team in 2023. So that was the first identity genetic linkage map for vanilla planifolia, which was obtained by genotyping self-fertilization population of our Reunionese clone. And one of the main information it gives us regarding the structure of the genome is that in fact we add the information of all the 16 pairs of chromosomes, with the chromosome 1 being split into 3 linkage groups. Well, what's next? Thanks to all these resources already available, and the one that we plan to improve, we hope to better decipher the molecular characteristic of partial endoreplication, as well as to work on really important questions for the vanilla sector, sustainability, such as the genomics of climatic adaptation in Madagascar leafless vanilla, as well as the molecular factors of the resistance to Fusarium oxysporum, and also the identification of molecular markers for several agronomic traits. So lastly, I'd like to thank all the people that collaborated on these works through the VANISEC consortium. Just remind you that there is this web platform. And well, thank you for your attention, and if you have any questions about this.
Thank you very much. We have time for questions. Do we have any? Oh.
Thank you very much for your talk. I just wonder, what are the methods in genomics to testing the resistance of Fusarium oxysporum in vanilla? For example,
well, we thanks to the genomic map and the genotyping of the population, we already, well, not me, but well, Félicien Favre and Quentin da Silva, that you can see here, already identified some genomic regions that could be involved in the resistance. So in the population that they genotyped, there were sensible and resistant varieties to this disease. In fact, in the population, in the self-population Sorry. In the in the self-fertilization population of vanilla planifolia, they found that some individuals already showed some resistance. And so they used this information with the genotyping and to identify this criteria.
I had one comment, and that was that I don't know if the photographs are the plants that you are actually analyzing. Are these the plants that you are analyzing when you?
No. We personally I just work on planifolia, and in the search of resistance, we also use some pompona individuals, I think.
Okay.
But all these plants, Aphyllus, vanilla, don't work on it personally.
It's just that I was just talking about the identities, and your second one, which you had labeled Mexicana, that's Colombiana. So just in case, if you're using that as Mexicana, then that's definitely
okay. I don't know.
Just correct the name. Yeah.
Thank you very much. Do we have any more questions? Last chance. I don't see, so thank you.
Thank you.